Good evening from South Carolina Governor School for Science and Mathematics. I'm Caspen Gregory, and today I'll be moderating the third presidential debate between President Donald Trump, former Vice President Joe Biden, and politician and academic Jo Jorgensen. This debate is sponsored by Dr. Jennifer Brown. During this debate, candidates will show off their knowledge in important topics in gene expression. Now, we will welcome the candidates to the stage. Good evening, candidates. We are here today to discuss chromatin structure, protein structure, types of DNA interactions, and types of DNA. Our Republican candidate, President Trump, will go first. President Trump, please tell us what you know about chromatin structure.
So, as you know, there are 3 levels of chromatin organization. The first level being the nucleosome, which forms the chromatin fiber. When they're unpacked, they form what look like beads on the string, which you can see in the figure. And these beads are the nucleosomes, and the string is the DNA. And they're composed of the linker DNA and nucleosome cores. Next would be the second level, which is the 30 nanometer fiber. It's made up of a bunch of nucleosomes that form a zigzag pattern, and the nucleosomes are pulled together by histone H1. Level 3. Level 3, here we have loop domains where non-histone chromosomal proteins create folded loops on the fiber. These loops are held together by clamp proteins.
Thank you, President Trump. Your minute is now up. Vice President Biden, your response?
Well, while President Trump mentioned the nucleosome, he didn't elaborate on what a nucleosome was. Even though it is not one of the 3 levels of chromatin structure, I think that knowledge is essential to understanding chromatin organization. The nucleosome is made of DNA wrapped 1.7 times around 8 histones, 2 of each, 2HA, 2HB, H3, and H4. Each histone has a tail sticking out.
Thank you. Now, Dr. Jorgensen, your response, please.
Adding on to Vice President Biden's point, I think it is important to mention how the histones are able to hold the nucleosome together. The histones are positively charged, so they are able to hold the negatively charged DNA together. If not for the histones, the DNA would repel itself and be unable to condense.
Thank you for that. Now we move to Vice President Biden. Vice President Biden, what can you tell us about chromatin remodeling complexes?
Chromatin remodeling complexes are protein complexes which use energy from ATP hydrolysis to move DNA. The energy released from the ATP is used to locally alter the nucleosome. Specifically, chromatin remodeling complexes use this energy to loosen the DNA wrapped around the core histones to make certain genes more accessible by changing the position of the DNA relative to the histone octamer. Chromatin remodeling complexes are not very active during mitosis because we need DNA to be as condensed as possible. However, they are necessary outside of mitosis because certain small but important genes might be inaccessible. So we need a specific protein to help them to make them available, similar to how I plan to make the funds available to increase access to education.
Vice President Biden, your minute is now up. President Trump, would you like to respond to Vice President Biden?
Well, chromatin remodeling complexes is just another thing that uses ATP. After all, everyone knows that ATP hydrolysis is essential for many functions in the cell. Maybe, Biden, you should increase your own access to education.
Thank you, President Trump. Now, Dr. Jorgensen.
Chromatin remodeling complexes are one way to locally alter the nucleosome, but there are others. Histone-modifying enzymes use chemical modifications to make DNA more or less accessible.
That's an excellent point, Dr. Jorgensen. Would you mind speaking on that?
Of course. Histone-modifying enzymes chemically modify histone tails, which regulate gene activity. These modifications are heritable, which is essential in helping cells maintain tissue identity because daughter cells need to know which genes their certain type of cell needs on and off. However, modifications are also reversible. So if you have a gene that was turned off during mitosis because it wasn't needed, but is needed during interphase, the modification which caused this gene to stop being expressed can be reversed. The histone modifying enzyme used to reverse these modifications would be an eraser, while histone modifying enzymes called writers add modifications and readers read modifications. Some common types of modifications are acetylation, which is adding an acetyl group and usually encourages gene expression, methylation, the adding of a methyl group, which usually...
Sorry to cut you short there, Dr. Jorgensen. Your time was up and the mute button was activated.
She got muted when she was talking about methylation, which mutes the genes.
Well, President Trump, it seems you've already responded to Dr. Jorgensen. So we will move right along to Vice President Biden. Vice President Biden, what is your response to Dr. Jorgensen?
Dr. Jorgensen forgot to mention that too much or too little activity of these enzymes can lead to harmful effects. If a tumor suppressor gene was deactivated through methylation, or if an oncogene was mistakenly turned on, that could lead to cancer.
Thank you for the respectful response, Vice President Biden. Anyway, moving right along. President Trump, could you tell us a bit about different types of DNA?
Why, yes. I think that there are many of us unaware of the diversity seen within DNA. The most basic idea that everyone has heard of is DNA would be bDNA. bDNA is the most commonly seen double-strand form that is depicted on models seen online, like you can see here. It has two unequal grooves, the major groove and the minor groove, also in the figure. I would also like to mention that the edges of the base pair within the DNA is exposed to these grooves. There is another conformation of DNA known as aDNA, which is a little less common to find than bDNA. Instead, it has 11 base pairs per turn, and the base pairs are placed relative to the DNA axis. Not to mention that its helix is shaped differently compared to bDNA. The major groove is much narrower and deeper compared to the major groove of bDNA, and the minor groove is much shallower than the minor groove. One thing to add is that aDNA has a right-handed helix. Thank you.
Thank you, President Trump. Vice President Biden, your response?
Now hold on there. What about the zDNA? Sure, zDNA has a more ragged appearance compared to aDNA and bDNA, but it is just as important as the other conformations. It plays a major role in gene expression and DNA processing events.
Thank you. And Dr. Jorgensen, would you like to add anything?
Yes, I would like to talk about zDNA structure. Unlike aDNA, it has a left-handed helix, meaning when looking at aDNA and zDNA, they appear to twist in different directions. The appearance of zDNA is caused by chemical modifications of cytosine, methylation, and high salt concentrations.
Thank you. And now we move to Vice President Biden. Vice President Biden, could you explain what you know about proteins?
Well, while DNA is very important, I feel that proteins are also something very important for the American people to understand. Like chromatin, proteins have different levels of organization. The primary level is the order of the amino acids of the peptide chain, the order which the DNA encodes the building blocks of proteins. The secondary level are the repeated regions of alpha helices and beta pleated sheets controlled by the hydrogen bonds within the amino acids. The tertiary structure are the other interactions between the amino acids, forming folded protein and giving it shape. Finally, some proteins have a fourth level organization, the quaternary structure, which are the interactions between one or more folded polypeptides to form a single unit.
And that concludes Vice President Biden's time. President Trump, would you like to give a response?
Yes, well, the structures of protein are important. I think you forgot to add why proteins fold in the shapes they do. It's all about minimizing free energy and forming a single stable conformation. America is so great. We have the lowest energy ever, the lowest free energy. just like a protein. And while some proteins do need a little extra help in the form of chaperone proteins, like you can see in the figure, most proteins are able to spontaneously fold into the lowest energy conformation as long as conditions are favorable. You wouldn't want to make it too hot, or your proteins might all unfold.
Thank you, President Trump. And Vice President Biden, your response?
Well, DNA and protein structure is fascinating. I think we should emphasize that these structures are very important because they influence DNA-protein interactions, which take part in many essential cell processes. The specificity of proteins allow for very specific and complex interactions, such as specific DNA sequence recognition.
Thank you for that, Vice President Biden. Now we have one more question left for tonight, so I'm going to ask Dr. Jorgensen, could you tell us about DNA protein interactions?
Of course, DNA protein interactions happen for a multitude of reasons. Some examples are for DNA replication or synthesis, DNA repair, transcription, and DNA coiling. Proteins bond to specific sites on DNA. These sites are characterized by functional groups such as H-bond donors, acceptors, and methyl groups. These groups are located on the edges of base pairs and help proteins recognize DNA sequences through chemical complementary.
And that concludes Dr. Jorgensen's time. President Trump, would you like to respond?
Yes, you forgot to mention that most of the functional groups used by proteins to identify DNA sequences do so in the major groove of the DNA, like you can see in the figure over here. Also, proteins rely on the major groove to bind because it is larger and has more variability, meaning it can be more unique to each sequence.
Thank you, President Trump. Vice President Biden, what is your response to Dr. Jorgensen?
It was also left out that the R groups are responsible for sequence recognition, which serves to facilitate the chemical complementary mentioned by Dr. Jorgensen, as well as that the shape of the proteins and grooves must be complementary for binding.
And thank you, Vice President Biden. Now that was our last question, so I would like to give the opportunity for each candidate to give closing remarks. Then this debate will conclude. President Trump, what would you like our audience to take from here today?
I hope the audience remembers the 3 levels of chromatin structure. We have the nucleosome, the 30 nanometer fiber, the loop domains. Also, don't forget the different types of DNA. It isn't all just one basic type you learned in introductory biology. There's B DNA, the typical DNA, the less common A DNA that has a narrower major groove as compared to B DNA. And finally, don't forget that there is also Z DNA, the left-handed version.
Thank you. And Vice President Biden, any final words for tonight?
Yes, I would like to remind the audience that chromatin remodeling complexes use ATP hydrolysis to shift the DNA as it was wrapped around the nucleosome. Additionally, protein structure is very important because it influences how proteins are able to interact with DNA. Proteins have 4 levels of structure: primary, secondary, tertiary, and sometimes quaternary. These interactions determine the shape of the protein and ultimately the function.
I'm sure Americans will keep that in mind. Now, Dr. Jorgensen, if you would like to state your closing remarks.
Yes, I would like the Americans to remember that histone modifying enzymes add reversible chemical modifications to histone tails. I would also like to remind them of the importance of DNA-protein interactions. Without it, we would not have DNA replication, DNA modification, and many other essential processes. The specificity of these DNA and protein interactions allow for amazing things and even sequence recognition.
Now, I would like to thank our 3 candidates for this very informative debate tonight, and I would like to thank South Carolina Governor School for Science and Mathematics and Dr. Jennifer Brown for making this debate possible. I hope you have a great night.